The chromatin fibers are visible in a cell which is not in its dividing condition. That means when it is in interphase or a resting phase, or once it is differentiated, at that time we find only chromatin fibers. As we are saying chromatin fibers, those are thread-like things. So it would be visible to us in the form of a thread. But when the cell enters a division phase, at that time these fibers condense, they become thick, and that is when we start seeing the chromosome-like structure. The chromosomes-like structure is thick, compact. So instead of a thread or a fiber, we start seeing a chromosome. This is chromatin fiber. What is in the chromosome? Each chromosome, if we enlarge this, each chromosome actually has two branches which are called chromatids. So these are the two chromatids. In the center there is a narrow constricted area, this is known as centromere, and this is a chromatid. Here we are talking about the two branches. So here there is this constricted area, and again these chromatids. In these chromatids run thread-like fibers. These fibers are known as chromonymata. And this constriction is the centromere. Centromere, because it is the first constriction, it is also known as primary constriction. In some chromosomes, other than this primary, there is one more constriction. So if we make that chromosome, it would look like the first constriction, that is the primary constriction, and there is one more constriction here. This is known as a satellite. So these chromosomes with the secondary constriction are known as SAT chromosomes. SAT chromosomes. In human beings, these SAT chromosomes are 13th, 14th, 15th, 21st and 22nd. So these 5 chromosomes are SAT chromosomes. That means they have this extra constriction, secondary constriction, known as the satellite. This region, which is the satellite region formed after the secondary constriction, is also known as NOR. NOR is nucleolar organization region. That means this is the region where nucleolus will be assembled or organized. So it could be a simple chromosome or it could be SAT chromosome. Simple chromosomes have only one constriction at the centromere, and SAT chromosomes have two. One is the primary and the other one is the secondary constriction, which helps in the formation of satellite. Now, on the basis of location of this centromere, we can classify these chromosomes into 4 categories. So these are the types on the basis of where the centromere is and how are the arms of these chromosomes or chromatids. So if this is the centromere and the two arms are of same length, then it is known as metacentric. Both the chromatids, their length is same, or we can say that the centromere is centrally placed. If one arm is little longer and the other arm is little shorter, then it is known as submetacentric. That means the centromere is not exactly in the center. One arm is little shorter and the other arm is little longer. So there is a difference in the length of these two arms. If one arm is very, very small and the other arm is very long, then such are known as acrocentric. And the fourth one is when one arm is totally absent. So there is a centromere and only one arm. Then such type of chromosomes are known as telocentric. English alphabets are used to represent these. Normally, this one is represented like this. Alphabet K. The submetacentric alphabet L. Acrocentric J. And telocentric I. So we can also see at some places I, J, L and K used to represent these four types of chromosomes based on the location of the centromere or the respective heights or lengths of the two chromatids or these two arms. So four types. Now let us see what exactly is this chromonemata made up of. It is made up of a substance called chromatin, and this chromatin is of two types based on its transcription capability. Euchromatin and heterochromatin. Euchromatin is an early transcribed chromatin. That means it goes early transcription in the S phase. So, early transcription. It shows early transcription. Early transcription, that means in S phase when the cell divides, at that time it can get transcribed. It is active. Active means whatever genetic material is present in this, it is going to help in protein synthesis. Heterochromatin is called late transcribing. Some part of it is inactive. On the basis of it, we divide it into two categories again. Facultative heterochromatin and constitutive heterochromatin. Both are the late transcribing. Facultative are the ones which are active for some time and then they become inactive. So, active for a stage and then become inactive permanently, but for some time they were active. In females, human females, there are 2 X chromosomes. One X chromosome after a certain stage changes into bar bodies. So, the non-functional X chromosome is the example of facultative heterochromatin. So, it is inactive after a certain stage. Constitutive, they are just structural, they are never active. So, they are never active. So, constitutive means because they are a part of our complete genome, but they are never going to get transcribed or translated into anything. Facultative, they are active only for a part of life and then they become non-functional. After that, they will never get active. And euchromatin is all the time active. So, whatever transcription, translation is taking place in our body is the euchromatin part, which is actually responsible for. Now, when we are talking of chromatin, these fibers, the chromonemata or chromatin fibers, when we are talking of, and we normally draw it in the form of a thread. If we magnify this, we see that it is not just a thread, there are some circular things which are seen in its structure. These circular things are actually nucleosomes. Nucleosomes are made up of histone proteins and DNA wrapped around it. So, let us talk about the nucleosome structure and we will see how these histone proteins and DNA are wrapped around to form this nucleosome. Nucleosomes are made up of basic histone proteins basic because they are basic in nature, DNA, one part of the DNA is wrapped around those histones and a part of DNA links to histones. So, we will talk about that, and there are some acidic non-histone proteins. So, basically there are main things, that is histone proteins and DNA. Histone proteins are 8 in number. So, there are 8 histone proteins which are present in the nucleus. These 8 are H2A, H2B, H3, H4. And all of them are in pairs. So, 2 H2As, 2 H2Bs, 2 H3s and 2 H4s. How are these histones arranged? These 2 which are in the front upper half are H2As. Behind these H2As, there are 2, that is, this one is behind this one and this. This is H2B, H2B. The lower half has H3 in pair, and behind them are H4. So, this is one and behind it. So, this is H4 and this is H4. So, total there are 8, and that is why this histone protein, 8 histone proteins collection is also known as octamer. It is called histone octamer. Around this octamer is a DNA strand. So, imagine there are 8 balls and which are to be tied up with one ribbon like thing. So, there is one DNA strand which goes over this, comes back behind it, again goes over it, so if we erase this inner part, what do we see? That this is the DNA which is going from here, back side, again coming from the front and again going behind it. So this DNA strand is wrapped around. But if there is nothing to hold this complete assembly together, then immediately everything is going to fall apart. So to hold all these things together, here at the top there is H1, that is histone protein again, H1, which is acting as a clamp to hold everything. So imagine as if you have 8 balls and you have wrapped it with a ribbon, and to hold that ribbon together there is a clip or a clamp. So that function is performed by this H1. Now this is one histone octamer and DNA wrapped around it. If you go back, what exactly is here? Again there are 8 histones. So again same arrangement, and this DNA, this is the same DNA which is continuing from here. So between 2 octamers, the DNA which is there is known as linker DNA. So one DNA which is the main one wrapped around the histone octamers, and between 2 histone octamers there is linker DNA. And that is why when we make this chromatin thread, it is not a straight line. At some intervals we find there are swellings, and these swellings are nothing but these histone octamers or nucleosome. So this complete structure is known as nucleosome. Nucleosome is also known as new body, NU body. A collection of 6 such nucleosomes is known as a solenoid. 6 make one solenoid. So histone octamers, which are basic histone proteins, and DNA. And these nucleosomes, when we say the chromatin fibers condense, all these structures come closer and it gives the shape of that chromosome. Solenoid, new body also another name given to this. So this is what is the nucleosome. Histone protein is the most important thing. These are basic in nature. So in the structure of nucleus, we have seen the nuclear membrane, structure in detail. We have also talked about the nucleoplasm. Then we saw the structure of the chromatin fibers and how they condense to form chromosomes. Apart from these 3 things, there is one more substance or structure which is found. It is known as the nuclear matrix. This nuclear matrix is nothing but protein fibers and these protein fibers connect nuclear membrane from one end to the other end. So if you are talking about just the nuclear membrane, these fibers are extending from one end to the other end. So they are providing strength to this nucleus as a structural part. So you can consider it as a cytoskeletal or similar to cytoskeletal structure. Now the only structure that remains which we need to discuss now is nucleolus. Nucleolus was discovered by Fontana. Number is variable. Normally each nucleus has one nucleolus, but again it is variable, so numbers can be 1 to 4 per nucleus. Nucleolus is going to help in assembly of ribosomes, so depending upon the condition of the cell. If the cell is actively involved in protein synthesis, more RNA and ribosome is to be synthesized, so it would have more number of nucleolus. But normally, if it is a normally acting cell, one nucleolus per nucleus per cell is what is present. It is without membrane. So there is no membrane which is surrounding this nucleolus. So then why do we draw it in a circular manner? It appears circular because there are fibrils. So there is fiber. These fibers, they are basically RNAs. And these fibers, they are arranged in such a manner that there are infoldings and outfoldings also. So these are RNA fibers. It is also known as pars fibrosa. And because of this kind of an arrangement, it appears to have a spherical shape. So just when we look at it, it appears as if there is a membrane around it, but actually this is not a membrane. These are RNA fibers. Then there is a matrix here, which is known as pars amorpha. Inside this, there are certain granules. These granules are the ribosomal units. These are ribosomal units or subunits. And there are some chromatin fibers. This is nucleolar chromatin. The chromatin fibers which are found in nucleus, they are known as nuclear chromatin. This is nucleolar chromatin. Nucleolar chromatin are a part of nucleolus. Now, where is this nucleolus formed? When we were talking about SAT chromosomes, that satellite part is known as NOR, that is nucleolar organizing region. So they are, this nucleolus, it is synthesized or formed in NOR. Nucleolus organization region, which is found in SAT chromosomes. So that is the place where nucleolus organization or assembly takes place. Function of nucleolus, it helps in ribosome formation, RNA synthesis. So that the main two functions, ribosome assembly or ribosome formation and RNA synthesis. So now with the structure of nucleolus, we have completed the cell part. Study of cell is known as cytology. So we have completed all structures which are inside the cell and the functions of all these structures. So basically we have completed the cell structure and functions. I hope you have understood each and every part. Just go through each and every section carefully and just keep revising so that each and every concept is clear.